In this video, we're going to continue talking about section 4.2 notes. We're going to review the principles of experimental design and go through each one in detail. So the four principles of experimental design are comparison, random assignment, control, and replication. Some of these words you've seen before in other branches of science. Some of them have slightly different meanings in the principles aspect. Let's look at first comparison. So comparison is the process where different treatments are performed within the same experiment in order to try and neutralize confounding. Remember, confounding is the thing that happens when you have two variables and you can't distinguish which has an effect on the response variable. This really only works when the groups are similar in nature. Bias will be introduced if groups of people aren't organized randomly. So if you have two groups that are fundamentally different in a big way, the comparison's not going to be as good as if the groups are very similar. So that way you can actually neutralize that bias. Which is why we use random assignment. Random assignment allows us to randomly pick which people get each treatment. So you can use technology, you can use numbers out of a hat, table D, whatever your personal choice is for random assignment. But you have to take the randomization out of the human's hands because humans are incapable of being random. We do this because it prevents bias from happening. Anytime that you choose or human chooses on their own without using some kind of random generator, number generator, or a random table D, there's bias in there, and even knowing about it's not good enough. You have to use that random assignment. Control. There are two reasons to control in the experiment. Number one, to prevent confounding. So in order to get rid of that idea behind certain variables having the same effect on the response variable, we control for those. For example, it may be like having two sets of groups of people controlling everything except for the treatments. So for example, the setting in which they take it, the people that are administering the tests or the treatments, and so on. So you prevent that confounding by controlling everything else. We also do this to reduce variability in the response variable. So this goes back to the confounding aspect. The variability is too big. It's kind of hard to describe from the explanatory variable. So control kind of gives it a nice window of possible values. All right, so replication. This is a word that's been used in other science classes, no doubt, but it means something different here. Replication in other areas of science means to be able to replicate or repeatable results of a experiment. So if person A in the United States has some experiment with these results, replication means someone in Europe or Asia or Africa can do the same experiment and repeat the same results. In the principles of experimental design, this means use enough subjects in order to weed out regular variation in the random assignment of treatment. So there's going to be a replication of the treatments more if you have more subjects. If you only have a certain number of subjects that's very small, you're going to have maybe too much variability to actually, or variation in the values and actually to make a conclusion. So replication means have enough subjects in order to make a fine conclusion or to actually look at the variation and make it a good summarization of the experiment. So it doesn't mean something different. Repeatable results and everything everywhere else. So it just means something different in this case. You're going to be able to describe a completely randomized design for an experiment, including how to randomly assign treatments using slips of paper, technology, or a table of random digits. Completely randomized design is the big phrase here. It's a way of designing an experiment that leads to the least amount of bias. So for example, let's say you're going to perform an experiment on 50 seniors at your high school, not including yourself. Describe in at least two ways to get a random collection of your 50 of your classmates. If you just want a random collection of classmates, you could simply do a simple random sample. You're going to choose 50, let's say, out of 200. It's a rough estimate. So that could be the table D. You could choose 3-digit numbers 50 times, going row by row. You could also do your random integer generator in your calculator. It's another way of doing it. You just need 50 integers between 0, 0, 1 and 200. And you're going to be choosing 50. If it's important for you to have, for example, boys, girls separated, you want an equal representation of each, you could do a stratified random sample. This is if you absolutely need all groups represented. And if you have the time to do so, because as we talked about before, stratified random samples take a little bit longer because you're doing multiple simple random samples. So you're repeating this process more than once. If you do a cluster sample, you can do that as well. Somehow get your senior class split up into clusters and then do a simple random sample of the clusters and take the entire cluster. Okay, so there's 3 ways, at least 2 ways. This is 3 ways to do it. Cluster samples really only work if you already have clusters there. If you try and manufacture clusters by yourself by making simple groups, that's kind of taking away the randomness of the actual clusters. So we want to make sure these are occurring naturally. So in the book, they review an experiment where they want to increase, or they want to test to see if the online course for the SAT prep class is actually increasing SAT scores. So what they do is they have 2 classes of 25 students. So that means they need 50 students total. So they get 50 student volunteers, and they don't come to the experimenters. The experimenters actually find 50 seniors and see if they would volunteer. So it's not a voluntary response. It's more of they go find the people and ask them to be a part of it. This part right here is the random assignment. That's the important part. Everybody has an equal chance of being in either group 1 or group 2. So that would be online or online or classroom treatment. If you don't do this randomly, you're going to introduce bias. You don't want to be picking people just by random, like what you might think is random standing and just picking random people in a row. Make it random using a number generator or using table D. So you have 2 groups of the same number. So that's part of your control. Make sure it's the same number of students for each. Okay. So part of your control, same number of students. Also, not mentioned in this diagram, same instructor. If you were to have 2 different instructors here, one for the online and one for the in-class, you might get some kind of difference, but you couldn't tell if it was from the instructor or from the actual online or in-class instruction. So make sure it's the same instructor. One also not mentioned here, same number of class sessions. If you were to have more class sessions on the online or the in-class, that may also introduce some confounding here, and you couldn't tell a difference between the number of class sessions or the actual instructor or the type of class. So you want to control all these things besides the treatment, and that way you can assess what's the difference between the 2 treatments overall. The last part here, comparison. You compare the 2 SAT scores. Look at their center, look at their spread. Is there a big difference? Is it statistically significant, which we'll talk about in the next video? And that's how you essentially do a completely randomized design. So sample your students, random assignment, control for everything but the treatments. So that could be number of students, that could be the instructor, could be the setting, number of actual students in the class. Compare at the end. In there also is the replication. Use enough students to where the data actually is going to help you decide whether or not there's a difference. If we instead used only like 4 students, that might not be enough. You need to replicate to kind of weed out that regular variation in the data set. So those are your 4 principles of experimental design. This is an example of completely randomized design. It's important in getting rid of that bias that we have, splitting up into 2 different treatments.